Our next is Andrew Field, who's currently a postdoc with Karen Endelman, another TCEPA awardee. Andrew's talk is going to be about enhancer transition from naive to primed embryonic stem cells. Andrew?
All right, thank you for having me on today. I'm excited to share some of the developments in the Edelman lab on our end of a collaboration that we have with Johanna Wasaka's group at Stanford. And we're picking apart the steps of enhancer assembly and assessing the potential role of nascent RNA in their function. So today I'll be outlining our approach and then talking about some of our preliminary efforts to narrow down in on some relevant time points to view enhancer assembly during differentiation. All right, so I'm sure this needs no introduction in this group, but gene expression dictates both the form and function of cells, and careful coordination of gene activation and repression is essential for differentiation development. And misregulation of this process, either during the establishment or maintenance of cell type specific programs, is often associated with diseases. We know that for a given chromosomal segment with two genes that might be proximal to each other, that genes may be active in different cell types. Turning on a gene in a specific or a cell type specific way often involves not just opening up the chromatin around the promoter, but also activating some of these distal intergenic regions that we're going to call enhancers. Different enhancers may be active in different cell types and thus conferring some of the cell type specificity of gene expression patterns that we see. Key to understanding cell identity and function is therefore in defining how these regions are selected and activated during cell differentiation and how they're maintained. So just opening up with some of our questions here. Want to probe how enhancers are selected and activated during differentiation. We want to decode the series of events that eventually lead to their activation. And then finally, assess the role of active transcription at enhancers and what functional role they might be contributing to the process. So let's break down some potential roles. There are many proposed models for the function of Pol2 at active enhancers, but there has not yet been any comprehensive analysis of the possible roles that RNA synthesis may play at these loci. So here we set up to distinguish between these models in a dynamic system. So first, maybe nothing's happening. Pol2 is just kind of opportunistically binding to these regions because of the open chromatin or the gathering of transcriptional machinery. But perhaps more interesting, Pol2 can be maintaining open chromatin by establishing a landing pad for transcription factors. Active transcription may also be stimulating the deposition of active chromatin marks in this region, perhaps through recruiting P300. Or finally, that nascent RNA itself may be involved in the stabilization of looping interactions, bringing the enhancer within close proximity of its target promoter. We have in our toolkit a number of inhibitors that can block the various stages of Pol2 function, including blocking the ability of Pol2 to bind, and thus inhibiting its ability to clear new chromatin. Inhibiting stable pausing of Pol2 to decrease its latency time at enhancers and thus affecting its ability to attract histone modification machinery. Or finally, roadblock blocking Pol2 elongation, not allowing it to actually process into active RNA synthesis, thus robbing it of its ability to create RNAs to participate in loop stabilization. We plan to read out at each of these steps by various sequencing techniques that you guys are all familiar with, including ATAC-seq, ChIP-seq, and capture-seq. This approach will allow us to detect any of these modes of activity in an unbiased fashion. Before we can really address the function of Pol2 and nascent RNAs and in enhancer function, we must first find a dynamic system in which enhancers are actively established and maintained, then identify the relevant time points when these key events are occurring. So for this purpose, we're focusing on the mouse embryonic stem cell to Epiblast-Like cell transition, or EpiLCs for short. These stages represent a kind of checkpoint that naturally occur over the first 2 days between the blastocyst formation and implantation in vivo in mouse. And we can take cells from each of these stages and grow them in vitro. Mouse embryonic stem cells kind of have this very tightly packed dome-like morphology when grown in culture, and EpiLCs look very drastically different, very spiky and flat in morphology. Both of these states actually retain pluripotency in that they can differentiate into any of the 3 adult cell types, but EpiLCs can no longer contribute to germline. While both of these states express key pluripotency factors, OCT4 and SOX2, it seems that OCT4 and SOX2 are redirected to new binding sites by OTX2 during this transition. And that's been shown in many beautiful studies before us. It has also been shown that many major imprinting events happen during this transition, including exon activation. And luckily for us, there's a way to culture these and initiate this transition in vitro. By plating ESCs on low density on fibronectin, withdrawing LIF, and adding FGF and serum, we can stimulate this highly synchronized transition to EpiLCs. Changes in morphology can clearly be seen in as little as 8 hours, where you start to have the spiky morphology start to pop up. And the cell continue to divide and mature, reaching completion by 48 hours. By gathering a tight time course of 1 to 2 hour intervals over this entire timeline, we hope to capture enhancers in the act of either being commissioned or decommissioned. All right, so how do you build an enhancer? So I'm going to display some of the general characteristics of what an enhancer is to us, or at least functionally defined. And the order may vary in this part of the presentation. But so how do you build an enhancer? Our current understanding is based on the functional components from static snapshots. So we hope to expand this by constructing a timeline of these events and how they're actually constructed in vivo. Our enhancer defining characteristics begin with open chromatin, which we can detect by ATAC-seq. And then active chromatin marks are laid down, including H3K27 acetylation and H3K4 mono and trimethylation. And they're also known to designate active enhancers as well as promoters. And we can detect these by ChIP-seq over this time course. Studies by us and others have identified widespread bidirectional transcription at enhancers, mostly producing these short, rapidly degraded eRNAs. And we have a variety of techniques to detect nascent RNA, and particularly I'll be talking about pro and grow-seq. And then steady state RNA can be measured by total RNA sequencing. Finally, enhancers have to convey their function through close 3D contact, which I'm sure we're all familiar with, and allowing for the interaction with promoters. And we hope to detect that by high ChIP and capture-seq. Altogether, these experiments will provide us with a complete picture of how enhancers assemble over time and give us a dynamic window and kinetic timeline to assess the individual functions of each of these components. So I'll be talking a little bit today about our preliminary grow-seq data and using total RNA sequencing to provide us with the bookends of the time window in which we need to dig deeper into some of these other techniques. So first, I'll talk about the grow-seq results. We and others have recently shown that the level of nascent RNA transcription correlates incredibly well with enhancer activity. So here we see these plots centered over functionally defined enhancers here, where we see start-seq reads, which is another nascent RNA sequencing technique that we've been using in the lab. And then sorting these peaks into quartiles by enhancer activity actually correlates incredibly well to the level of transcription that we see by start-seq. In fact, about 95% of functionally defined Drosophila enhancers generate these short nascent RNAs, and we've seen it replicated to a similar degree in mammalian cells as well. We can use this property to identify enhancers and assess their potential levels during differentiation. So here I'm showing a genome browser shot of Correlates data with the positive strand reads in red and the negative strand reads in blue. In this region, just upstream of OCT4, we have two previously identified enhancers that are expressed in embryonic stem cells, highlighted in blue. And you'll notice that at each of these enhancers, you have this very typical bidirectional expression pattern detected by Grossi. And these these overlap peaks that we see in H3K27 acetylation. Charles Danko has developed a tool called DREG that takes advantage of this bidirectional pattern in order to call peaks that are likely to be either promoters or enhancers. And I've been using this tool in order to call potentially novel regions in here, and using the rebuild up on each of those locations in order to give us an idea of how active each of these enhancers are. So I would like to walk through a couple examples of what we've seen with our preliminary Grossi data. And first on this list is the BRAF locus, which is an essential developmental gene that's often misregulated in cancers. While expressed at low levels in ES cells, we see it's stimulated by 12 hours in our Grossi data, alongside two putative enhancer elements highlighted in yellow here and here. While the distal enhancer tracks well with the activity of the promoter in the region, the proximal enhancer loci spikes really early and then it's virtually gone by 24 hours. So we see this transient expression pattern, and I'll be talking a little bit more about that later as well. Another essential gene involved in differentiation is the polycomb-associated JARED2. And it's actually fairly highly expressed in embryonic stem cells, but it undergoes a boost during this transition. And we see that also correlates with these two potential enhancers upstream of it as well, where, again, the more distal enhancer seems to have this transient pattern where it boosts on early and then is off by 48 hours. But then we have this other one that's turned on and maintained throughout this process. So we think that this is actually maintaining the signal that's shared with JARED2 here. And then finally, there's the OCT4 locus. So these are two previously identified enhancers that I've mentioned before. We have the proximal super enhancer that's been very well studied in the system. And OCT4 doesn't change transcription over this time course, even though by Grossi you'd see this very large spike very early on that eventually peters out. But I'll later show RNA-seq data that shows that the actual steady state level of this gene stays the same. But even still, you see the distal enhancer, which seems to be the most active in the ES cell state, is actually diminishing to almost zero at the 48-hour time point, where it seems to be compensated by the OCT4 super enhancer during this transition. And that one seems to take over the control of the OCT4 locus. So taking the data as a whole, I can break down enhancers into observed groups. So first we have the ES cell associated peaks, where we have them mostly expressed in ES cells and then they peter off by the end. We have the EpiLC associated peaks that are turning on over this process and getting established. And then finally, there's a large group of transiently expressed potential enhancer units that seem to be turning on early and then turning off during this transition. An overwhelming 59% of the detected putative enhancers are differentially active during differentiation by Grossi reads, despite a relatively minor steady state gene expression changes, which I'll talk about a little bit later. Of those, 30% are newly established in the EpiLC state, shown here. And then over 20% are only present in transition states, which was kind of surprising. Perhaps more surprising is that over 2,000 loci undergo enhancer switching, similar to the OCT4 locus. A large proportion of these exhibit little or no change in the expression of their neighboring genes, which we think are being affected by them. So it's an enticing question to think why this is so prevalent, and are they just redundant enhancers that are activated with the presence of OTX2, or maybe there's new enhancers that are more labile or labile than their predecessors in order to prepare the cells for further differentiation. Or they may be acting on more distal gene targets that we're not looking at directly with just the Grosseck reads. But getting back to our original question, we really want, we still have many differentially expressed genes that occur alongside a lot of these enhancer activation events. So in order to really put bookends on this process and identify the optimal time windows in which to observe enhancer assembly at these loci, I compiled a tight time course of total RNA-seq over this differentiation. So looking at the examples from before, we see the effect of enhancer dynamics on the steady state gene expression levels. So in black here, we have the RNA-seq, and then in blue, we have the proximal enhancer, and then red, the more distal enhancer at each of these loci. And starting with BRAF, we see the peak of the proximal enhancer activity come on early, even before the gene really seems to be boosted all that much. And it doesn't seem to have much of an effect on global gene expression. But as the more distal enhancer becomes established, we see a nice correlation with the rise in steady state of the gene expression. JARED2, on the other hand, has a very different kinetic profile. Both enhancers come on very early and very strong, leading to a quick and immediate boost in gene expression that we see by 4 hours. As the proximal enhancer is established, we see a drop in the distal enhancer. So it seems like the proximal enhancer is taking over in this case. And then finally, we have the OCT4 locus, which is steady as a rock throughout the whole process. But it still seems to have this kind of baton handoff between the two enhancers, where you have one being decommissioned and the other one coming in to replace it. So looking at the RNA-seq data set globally, I wanted to identify the timescale on which we have the largest changes in gene expression. On the left, we see a PCA plot here, and we see that it tracks very nicely over time for the most part, but then there's this big gap between the 0 and the 4-hour time point that we're associating with initiation. We also see that mirrored in the DE-seq determined distance between the samples, where we see the largest difference within the first 12 hours is actually due to this 0 to 4-hour time point. So we know that we're going to need to more heavily hit this very early time point. So in summary, I'd like to wrap up that we have this very synchronous system of mouse ES stem cells to EpiLC transition, and we have that working very well in our conditions. Using that technique, we were able to, along with nascent RNA sequencing techniques, identify active potential enhancers that are ESC associated, EpiLC associated, and actually transient as well. And among those, perhaps surprisingly, we see a lot of these enhancer switching events that aren't necessarily coupled to the most proximal genes. So we're really interested into digging a little bit further into what those guys might be doing. And then finally, after we establish this timeline and using all of our techniques, we can pick apart the series of events that actually lead to enhancer activation and decommission. And then finally, try to break it by introducing our inhibitors for Pol2 function and see what it might actually be doing in this process. So with that, I would like to thank the 40N Nucleome project for funding us to have this really comprehensive approach to gene regulation. Karen has been an excellent mentor, and I really appreciate her support. Telmo Henriquez has done a lot of preliminary work establishing much of the basis of this project. And Eilina Blondi is a graduate student who's working alongside me on the grant. And then finally, Johanna Wasaka's group. They've been an excellent collaborative partner, providing us with cell culture protocol support, precious samples and just been excellent all around. And with that, I'll take any questions.
Thank you, Andrew. This is very clear and exciting work. Any questions? We have about 10 minutes.